Mastitis is a complex, multi-faceted disease and is one of the most expensive dairy cow health problems. It also affects dairy processors, as it results in reduced product quality, cheese yields, and shelf life of dairy products. Previous research, conducted under Dairy Research Cluster 2 by the Mastitis Network, estimated the losses associated with mastitis for Canadian dairy producers at $665 million per year, which represents 10% of net farm receipts in Canada. An interdisciplinary team of Canadian scientists and trainees was assembled into a coordinated research and knowledge transfer program to make a significant contribution through research to the sustainable production of safe and high-quality milk. The network vision will be directed into 3 research themes: animal, pathogen, and the environment. 9 coordinated research projects will be conducted over the 5-year period, and they are grouped under each research theme. So let's discover them. The first theme is the animal theme, and the first project in this category is about understanding immune suppression following calving. The aim of this research project is to reduce disease incidence and antibiotic utilization by stimulating cow immunity. The principal investigators for this project are Pierre Lacasse, Agriculture and Agri-Food Canada, Xin Xiao, McGill University, and Richard Bloin from the University of Sherbrooke. The second project is entitled Cow Defense Peptides, a New Treatment Alternative. It aims to develop therapeutic alternatives and genetic selection tools to control mastitis. The principal investigators for this project are Eduardo Cobo and Herman Barkema from the University of Calgary, Robert Hancock from the University of British Columbia, Graham Plasto from the University of Alberta, and Marcus Colazzo from Alberta Agriculture and Forestry. The third project is entitled Polyvalent Vaccine Against Bovine and Mastitis Pathogens. The goal of this project is to develop a polyvalent vaccine against multiple bovine and mastitis pathogens. The principal investigators for this project are François Malouin from the University of Sherbrooke, Pierre Lacasse from Agriculture and Agri-Food Canada, and Marv J. Miller from the University of Notre Dame. Let's now discover the pathogen theme. And the first project in this second category is about understanding factors leading to adhesion of Staphylococcus aureus in the udder. The aim of this project is to characterize the attachment and the biofilm production by Staph aureus isolates into mammary epithelial cells. The principal investigators for this project are Xin Xiao from McGill University, Mario Jacques from the University of Montreal, and Pierre Lacasse from Agriculture and Agri-Foods Canada. The second project is about developing a fast and accurate tool for predicting resistance to antimicrobials by mastitis-associated bacterial species. This is done by developing a MALDI-TOF beta-lactamase assay for rapid, cost-effective, and accurate determination of beta-lactam resistance of Staph aureus isolates. The principal investigators for this project are Greg Keefe, J. Trenton McClure, and Ibrahim El-Sohabi from the University of Prince Edward Island, Herman Barkema and Vineet Saini from the University of Calgary, and Simon Dufault from the University of Montreal. The third project is entitled Impact of Staphylococci Intramammary Infections on Quarter Milk Yield and Milk Composition. The goal of this project is to elucidate the effect on quarter milk yield and composition of the most common non-aureus Staphylococci intramammary infections. The principal investigators for this project are Simon Dufault, Jean-Philippe Roy from the University of Montreal, Rachel Gervais from Laval University, Trevor DeVries from the University of Guelph, Pamela Adkins and John R. Middleton from the University of Missouri, and Greg Keefe from the University of Prince Edward Island. The fourth project is about understanding links between the bovine milk microbiome and mastitis. And the aim of this project is to characterize changes that occur in the milk microbiome. The principal investigators for this project are Jennifer Ronholm and Lawrence Goodridge from McGill University, Min Lin from the Canadian Food Inspection Agency, and Debra Sanchi from Lactanet. Let's now move on to the environmental theme. And the first project in this third category concerns the impact of dry-off management in automatic milking systems on udder health. The objective of this project is to determine how milking frequency, feed allocation, and milk yield variation in the weeks leading up to dry-off in robotic milk cows will affect the risk of intramammary infections after calving. The principal investigators for this project are Trevor de Vries from the University of Guelph, Simon Dufault from the University of Montreal, and Herman Barkema from the University of Calgary. The second and last project in this category concerns the impact of applying an on-farm mastitis intervention program. The objective is the adoption of the best management practices concerning udder health. The principal investigators for this project are Christopher D. Luby from the University of Saskatchewan, Jean-Philippe Roy and Simon Dufault from the University of Montreal, Herman Barkema from the University of Calgary, and David Kelton from the University of Guelph. So, as you can see, through these projects, the network seeks to provide knowledge enabling the industry in order to further decrease the national average somatic cell count, decrease losses from culling due to mastitis, optimize best management husbandry practices and antimicrobial use, and finally, to characterize on-farm control points that will support dairy farmers' ability to produce the high-quality Canadian milk that we're used to and to protect public health. What an exciting program! Follow us to find out the results.